Good morning, students. Welcome to our new module. It's called Basics of Preclinical Studies, and today's topic is mostly introduction part. And here we have a lecture plan, and we will talk about definition of physiology and the development stages of physiology. Then we will talk about physiological research methods, and then some concepts about function regulations. Here the definition is given by it said physiology. It's physis, it's Latin word, it means nature, and logos, it means science, combined with these two words, and studying of nature kinds of. And actually it said here, it's science which is studying function of living organisms on or systems or bodies or cells or organs. And again it said it's also studies the regulation function of bodies. And again here it said physiology considers organism functions in an interconnection with environment. In words, I will say physiology is defined as a study of functions of various systems and different organs of the body. And we have different types of physiology. We have a human physiology, and we have animal physiology, and sometimes we have plant physiology. And now we are just mostly focusing on our human physiology. And the human physiology, it will connect with biochemistry, pharmacology, and pathology, and we could say it connects with all of these subjects which you will learn next year. And again, if you want to study physiology, you must know the knowledge about anatomy. Mostly is anatomy, because anatomy and histology is connected each other. It said physiology belongs to biological science. It's very obvious. And it said it's closely connected with anatomy, histology, chemistry, and physics, etc. And again, here we have development stages of physiology. In our book or in our university, we are just divided it into 4 stages. So stage number 1, it's called pre-Harvey stage, and stage number 2, it's called Harvey stage, and then you have Pavlov stage, then you have modern stage. Pre-Harvey, it starts from the Hippocrates. Hippocrates in Western country, it's considered as a founder of medicine. And then Harvey, William Harvey, it's a British scientist, and it has a very famous kind of book. It's called Circulation of Blood. And this person's just not only focusing on the anatomy, because Harvey start investigating bodies connect with physiology, and he's studying anatomy, also studying physiology. Before Harvey, anatomy just focused on the discovery and the description of the organ without investigating the organ's purposes or uses in the body. But after Harvey, and just start from Harvey, Harvey just focused on the both sides of the anatomy, not only focused on the description but also focused on its functions. And again, Pavlov, it's a Russian scientist, developed a surgical method of chronic experiment with extensive use of fistula. Fistula is tube, and which enables the functions of various organs to be observed continuously under relatively normal conditions. And because of his discovery, and he considered as opened a new era in the development of physiology, and until he developed these chronic methods, The principal method used has been that acute vivisection, and the function of an organism had only been arrived by a process of analysis. Said modern stage, and here it said neurocybernetics, and because of its modern neurocybernetics, it's kind of artificial intelligence. And then we have some methods of physiological research. We have mostly two methods actually. One is acute, one is chronic methods. And it's said acute is very quick, and chronic is just prolonged, slow methods. And in the acute methods we get results immediately, and the chronic methods we get results after some time, maybe 1 week, 1 month. And the chronic methods, you must know, it's developed by the Russian scientist Pavlov. And here we have a vivisection in the acute methods and studying of isolated bodies, and we could say actually it's experiment in vitro and experiment in vivo. And again here we have third types of methods. It said clinical physiological methods. It is methods studying patients in the hospital. And then we have some regulation of functions of an organism. Mostly we have two regulation functions systems. One is called neuroregulation, and then we have second is called humoral regulation. And it's said neuroregulation, and it's based on the reflex activity. And reflex activity, it has 5 components. It starts from receptor, afferent neurons, center, and then efferent neuron, and we have effector organs. And humoral regulation, it is at the participation of chemicals in the circulating in the blood. It's called hormones. And here neuroregulation is very, very quick and accurate, and however humoral regulation is smaller than neuroregulation. And sometimes we have neurohumoral regulations. It's very, very important. And again we have autoregulations. Autoregulations, it is not depending on neuro and humoral regulations. Sometimes cells can regulate its function by itself. And again here we comes from neurohumoral regulations, and we are using nerve system and the endocrine system together, for example to water and to maintain the water. And again in the physiology we have one important concept. It's called homeostasis. Homeostasis, it is maintenance of constant internal environment of the body. And the internal environment, it's extracellular fluid. We have blood, it can considered as extracellular fluid. And the functioning of homeostatic mechanism, the body must recognize the deviation of any physiological activities from the normal limits. Fortunately, body is provided with appropriate detectors. We call it receptors, which recognize the deviation. Then these receptors sense the deviation and sends alert to the centers by afferent neurons. And in the center it will analyze and integrating the information, and again then it immediately sends information to the some related or considered effector organs. And then we either accelerate or inhibit activity of that mechanisms, so that we can maintain our body functions. We have components of homeostatic systems. We said detectors and receptors, or we have control centers, and we have effectors. We have nerve fibers. We have afferent and efferent nerve fibers. And again, in our body, we have a feedback system, and we have negative and positive feedback system. A negative feedback is one that react in a way to arrest or to stop the change or reverse the direction of that change. After receiving a message, effectors send negative feedback signals back to the system. Then the system stabilizes its own functions and make an attempt to maintain homeostasis. Example of negative feedback is to maintain body temperature, to maintain pH. And again, we have positive feedback. It's a system which reacts in a way to increase the intensity of the change in the same directions. And you know, in our body, positive feedback is less common than negative feedback. And however, it has its own significance during emergency conditions. For example, we have a blood clotting system. This system is actually its positive feedback system. And again, one more example is to give birth. It is a positive feedback system. Example of positive feedback system. And now for today, it's